spontaneous cessation of hemorrhage when a blood vessel get injured. So when a blood vessel get injured, there will be bleeding. So the mechanism that stops the bleeding is called hemostasis. It is hemostasis. When blood comes out of the blood vessel, it is the vessel, the injured part is healed so that no more passage of blood from the vessel. So it is done by hemostasis. So what is there in this process? Events. Try to remember. What are the events taking place during hemostasis? This is a blood vessel injured. Blood is coming out and spontaneously there will be cessation of bleeding. This is hemostasis. Now, what is the phenomena going on? Immediately, because of injury to the blood vessels, spasm in the blood vessels. There will be spasm in the blood vessels, which is created locally. Local spasm will be there. So this blood vessel will be narrowed because of the spasm. As a result, less blood will be flowing. See, this is one of the protective mechanism through spasm, originated by because of injury, local injury. And there are some pain receptors in the injured area. They will be stimulated. This stimulation will go to hypothalamus, to the brain, and the brain will cause reflex vasoconstriction throughout the body, particularly the peripheral region. Okay, so again this will cause narrowing of the blood vessel. As a result, there will be further reduction in blood flow. Moreover, during this process, we know the platelets, we have already discussed, platelets have some granules, dense granules, alpha granules, and when these platelets adhere to the injured tissues, liberate those granules, and within these granules, some of the granules are 5-hydroxytryptamine, histamine, they causes vasoconstriction. So again third factor is coming. They will also initiate vasoconstriction. Okay, so this is the initial phase of vasoconstriction. Then the platelets will come into action. This wall, cross section of this wall, this is the lumen. There are how many layers? 3 layers. Innermost layer is endothelial layer, made up of endothelial cells. And just outside the endothelial cells, so this is the, and these are the endothelial cells. Just outside the endothelial cells, you will find collagen. You will find collagen. These are connective tissue. So this collagen fiber, when the platelets, so here the platelets will come in contact with the collagen fibers. The platelets are having binding receptors, which we know we have studied, there are different type of receptors. There are receptors to bind these collagen fibers. There are receptors to bind Fon, Willeward. So these things will bind with the, when this is exposed, vessel is exposed, so this thing will bind. And some of the platelets will attach to the collagen fibers. And this will stimulate the platelets to become activated. When these platelets are activated, they will form like pseudopodia-like structures. Okay, and at the same time they will liberate from the granules some ADP like that. So that will attract other platelets to adhere there, to aggregate there. So as a result, there will be aggregation of so many platelets here. And these platelets will form a plug. This is the temporary platelet plug. This is not permanent. Even after this formation of plug, blood may come out. So it has to be made permanent. It has to be stabilized. So it is done by the next process, means coagulation. So before coagulation starts, actually, there is formation of platelet plug. Okay, understood? So now, let's see what is coagulation. We know what is hemostasis. So coagulation is a part of hemostasis. And formation of platelet plug is a part of hemostasis. Vasoconstriction is a part of hemostasis. They all of these contributing the cessation of blood flow through the injured area. Then what is coagulation? Coagulation is the process. It is the process by which the blood is converted into jelly-like semi-solid structure. Okay, this is called clot, and because the formation of clot by this process, there will be cessation of bleeding. Okay, there will be cessation. So this is coagulation. Coagulation or clotting is defined as a process in which blood loses its fluidity and becomes a jelly-like mass within a few minutes after it is shed out of blood vessel. Okay, this is coagulation. Is that clear? What is coagulation? I will ask. Try to remember what is coagulation. Now there are so many factors involved with coagulation. There are 13 factors. The first factor is what? Fibrinogen. Second, prothrombin. Third, tissue thromboplastin. Fourth, calcium. Fifth, except tissue thromboplastin or tissue factor and calcium, there are 13 factors. Except these two factors, all are synthesized in liver. All are synthesized, remember this, all are synthesized in liver. And then calcium, labile factor, and then presence has not been proved. Factor, which one? Which factor? Factor 6. So actually there is no factor 6. Factor 7, the stable factor. Factor 8, anti-hemophilic factor. Means this factor prevents hemophilia. We will study hemophilia are of two types. Hemophilia A and hemophilia B. So this is when this factor 7, factor 8 is absent, that condition is called hemophilia B. Okay, and out of total hemophilia patient, only 50% suffer hemophilia B, means because of deficiency of this factor. And there is factor 9 is plasma thromboplastin component. Okay, deficiency of this factor also may cause a disease. We'll discuss later. Then Stewart factor, this is important, very important, because it is another hemophilia, that is hemophilia A. It is most prevalent among all the hemophilic patients. 85% belongs to this group. Okay. This disease was prevalent in royal family of England. You know that? Okay. So all these are factors. So you have to learn. You have to learn these factors. Okay? Those who are Bengali, I'll give you an easy method to remember serially. Not now, because I don't know how to translate this in other language. So that's why I'm not telling in the class. If possible, you do transfer. So
when will you get it?
Huh?
When will you get it? We will learn the Bengali.
Okay, I'll not now. Okay. Mechanism of blood coagulation. There are 3 phases. The first phase, what will be there? There will be formation of prothrombin activator. This prothrombin activator is common. Intrinsic pathway will also produce this. Extrinsic pathway will also produce this. So this may come in first phase, the end product of first phase may come either from extrinsic pathway or from intrinsic pathway. Which one is faster? Which one is faster? No idea. The intrinsic path is faster. Okay, so first IPA. IPA stands for intrinsic prothrombin activator. EPA means extrinsic prothrombin activator. So there are 2 sources. Okay, then formation of thrombin to thrombin. And formation of thrombin. Thrombin is an enzyme. And prothrombin these are proenzymes. This is proenzyme synthesized in the liver. And finally, the final stage, formation of fibrin from fibrinogen. So these are the 3 main steps. The factors, most of these factors are actually proenzymes. So they will be, when they are activated, they form enzymes. So actually, one inactivated enzyme, when activated, becomes activated, and this enzyme will activate another proenzyme into activated form. This will activate another proenzyme into. So there is a cascade phenomena, waterfall phenomena. Either we call it as a cascade phenomena or waterfall phenomena. So these are the things, as I said, steps of blood clotting. Repeated. Intrinsic path, extrinsic path. So these are here actually steps are described. I will come into this. So this will be easier. So you see, this is which pathway? Intrinsic pathway. This is intrinsic pathway. These are the collagen fibers. Okay. The fact, I will start with intrinsic pathway. So blood will try to come, and this blood is having plasma, and thus that plasma will contain different factors. One of the factor, that is factor 12. When it comes in contact with the broken tissue, it becomes activated. See, factor 12 inactive, it is become activated when they made contact with collagen. And some other factors help this process. This is kallikrein, HMG kininogen, and positive surface of the endothelium. Negative surface of the endothelium. Endothelial cells, the luminal surface are actually negatively charged. And the RBC's outer surface also negatively charged. So they repel each other. As a result, they cannot come in contact. So what happens? They always remain in suspension. They cannot adhere. If there is adherence, there the process of coagulation will start. So this one now activated, and this one will, as enzyme, this is an enzyme, now it will inactive 11 will be converted into active 11. Again, this will catalyze the conversion of this into this. This is the axim is activated 10A. What is the name of the factor 10? What is the name of the factor 10? So this factor will be activated. So this is the actually tissue. There is another name for this, tissue thromboplastin. Okay, so this will be prothrombin activator. Not this is prothrombin activator. So this is actually prothrombin activator. Came from intrinsic pathway. Okay, let's see how this thing comes from extrinsic pathway. This is the extrinsic pathway. Here what happens when the tissue is damaged, one other phospholipid and lipolipid come out of the damaged cells. And these are called tissue thromboplastin. These are actually component of the injured cell. When factor 7 comes in contact with this tissue thromboplastin, this is actually factor 3. Now this factor 3 will help conversion of inactive 7 into active 7. Now active 7A will convert inactive factor 10 into active factor 10. So by this way, from two sources we got 10A. Two sources, one is through extrinsic path, one is from intrinsic path. Now let's see what they do. What this 10A does. Active 10A with the help of factor 3, calcium ion and platelet phospholipid, this will be converted into, this will stimulate prothrombin into thrombin enzyme. Now thrombin is an enzyme in presence of factor 13 and calcium ion will convert fibrinogen into fibrin. Fibrinogen, these are made up of polypeptide chains. Numbers of polypeptide chains. So what thrombin does? It actually breaks the peptide bonds that connect each of the peptide bonds to one another. So first they will break this. As a result, all the monomers will be freed. Okay? So initially, thrombin will cause formation of fibrin monomer. Now these fibrin monomers will join to each other and will form a thread. And this is fibrin polymer. This fibrin polymer is soluble. Now factor 13 converts this polymer into insoluble polymer. Done? Now it is insoluble. Now it will form a meshwork. Now it will form a meshwork, network. And within this net, the cells, which cells? Because platelets are already there, it is happening in the injured area only. All these changes are happening in the injured area, in the cut area. Here, interesting, actually all the reactions are going on here only. So here already platelet plug is there. And there are also some lot of fibrinogens are there. Now they have converted into fibrin mesh. So this fibrin mesh, this was the platelet. When they came adhered to the collagen fiber of the endothelium, they became activated. Each of the platelets became activated and they started forming spicules. With these spicules, they will find other thrombocytes, other platelets. So they will come together, they will join together to form the plug. Now this fibrin mesh, this is the spicule of another thrombocyte. Now the fibrin will be tugged with these spicules. So now when we know the spicules are having actin myosin, we learned previously. So this actin myosin will contract. So spicules will be contracted. As a result, the fibrin will also be contracted. The whole the clot is already formed when RBC platelets were trapped within the mesh. So now it will undergo retraction. How retraction happens? Because of shortening of the spicules. Because the spicules are having actin and myosins. With the help of the spicules, all the platelets coming together and the mesh work also coming closer. So within this mesh, within this clot, there are some fluids. So this fluid will be coming out of the clot, that is serum. That is nothing but serum. So now the clot is solid, almost solid. So it is formed a solid clot there. Okay, it is not loose now. So now blood cannot pass through. Cannot. So this is the retraction. This is clot retraction. Now, why there is retraction? What is the need? Because the injured area has to be repaired. So I need the space. So from both ends, there will be, under the influence of growth factors, both factors will start multiplying. The cells of the injured tissue will start multiplying. So they will grow and will be coming closer. So before that, the clot has to be eliminated. Before they come together, clot has to be eliminated, and this is done by fibrinolysis. Now the fibrinolysis will take place. If this clot enters into the lumen, what will happen? It will travel through the blood, and ultimately what it will cause? Blockage of the blood vessels. So suppose one of the blood vessels coming to my hand, it is blocked by this clot. So what will happen to my hand? It will not get blood, it will not get oxygen, it will not get nutrients. So my hands will be necrosed. So to prevent that, it has to be splitted. The clot has to be broken into small pieces. And these are called embolas. These are called embolas. So these embolas we can find when there is intravascular hemorrhage. These embolas can appear in lung, in kidney, and if they block the blood vessels, the area of lung will be necrosed. Area of kidney will be necrosed. Area of spleen will be necrosed. So that it cannot happen. So there are some processes. Another example of intravascular coagulation, atherosclerosis. Atherosclerosis, what happens? There is lipid accumulation in the endothelial cells. As a result, inner lining of endothelium becomes rough. Inner lining becomes rough. So when blood flows through this rough surface, it will be, the platelets will be broken and coagulation will be initiated and clot will be formed. Now this clot, by the internal mechanism of the endothelium and liver, this will be converted into pieces under normal condition. Then they will be phagocytosed. But when its quantity is much more, this ability is not sufficient. Then there will be actually more formation of clots. Now they will be converted into embolas and will be blocking some blood vessels. The process is called emboli formation. Injury, platelets are coming, forming spicules on these fibrins. These are the fibrin attaching the platelets and aggregation of platelets. These are the factors. This is what? Extrinsic path. Inactive 10. This is inactive 10. Now it is converted into active 10A. This 10A came from extrinsic source. Now this is the intrinsic path. Started with 12. Then it converted 11A. 11A. Now 11A will convert 10 into 10A with the help of the 8A. Prothrombin, and one molecule of 10A may convert about 1000 molecules of prothrombin into thrombin. May convert one molecule of this 10A can convert about 1000 molecules of Now see, these are the fibrin threads are producing. Fibrin threads, polymer. The mesh network. So all this is clot. Hello, one question for all. So you can see within this clot there is only platelets and RBCs. But WBCs are bigger in size in respect to platelets and RBC. Then what they are? They will avoid the mesh. Is it? Out of all the students, if one student doesn't come, then what does it mean? It doesn't mean anything. All are present. So, absence of one is negligible. See the quantity of WBCs and the quantity of RBCs and platelets, very negligible. That's why we are ignoring its presence. This will, presence or absence will not make any difference. Understood? Clear to you? Clot retraction already discussed for 4 points, 4 marks, important. And this is fibrinolysis. How fibrinolysis actually happens. This is the fibrin in the clot. We are not disturbing RBC or platelets. We will be disturbing only the fibrin threads. We will cut it so that all the parts become separate. Okay. So what is this is the fibrin is cut into fibrin degraded products. Who did it? Plasmin. Plasmin normally remains inactive in blood, in plasma. This is activated by plasma. This comes from plasminogen. So this is activated by tissue thromboplastin activator and urokinase. These are present. These are present. Even these are present in platelets. So they will convert plasminogen into plasmin. Now this plasmin enzyme will cut the fibrin into pieces. So there will be fibrinolysis. This is the mechanism. You have to explain what is the mechanism. Fibrinolysis. Are there any importance of fibrinolysis? Yes, clot will be broken down. Otherwise, what can happen? This blood can pass through the coronary circulation, coronary blood vessels. There it may cause blockage. So there will be no passage of blood, no supply of oxygen to the area which is supplied by that blood vessel. So that vessel, that area will not get any nutrient oxygen. So it will suffer ischemia. Initial sign symptom. Then when there is total blockage, then it is called heart attack. Then if it is a large area, then with the single attack, the patient may die. If it is a small area, then it may be recovered. When the patient is admitted to hospital, immediately some drug, anticoagulant is given. Some anticoagulant is given so that these are hemolyzed. There is fibrinolysis. So this clot will be converted into small pieces and will be eliminated. Okay, so it may happen in cerebral circulation. So it may block some blood vessels of cerebral circulation and cause stroke. Similarly, it may cause obstruction of pulmonary blood vessels. So it may affect any part of our body. That's why normally it should be fibrinolyzed. The clot has to be fibrinolyzed. Now tell me, this question may come, very important. For university exam, internal exam, Viva Voce, this is important. What is the question? Simple question. So you have to answer. You will be telling, sir, our blood is having heparin. Not only heparin. There are some other factors. What are these? Protein C and the plasmin we say. If there is any formation of clots, immediately this will be broken. By chance, if any clot is formed, immediately this will be broken by the plasmin. So this will prevent intravascular coagulation and endothelial factor. Firstly, the lining is smooth, there is no chances of breakdown of the platelets. I said the platelets are fragile. So if it is smooth, no. If it is rough, then it will undergo breakdown. And the process of clot will be started. And what is this? An endothelial factor, smooth lining, and this lining is negatively charged. Whereas plasma factors that supports, that causes coagulation, they are also negatively charged. So lining is negatively charged and the factors are also negatively charged. So they cannot come closer, they will repel each other. So this is another factor. And velocity of circulation. Blood is flowing. When blood flow is less, there is formation of clot.